So thank you all for joining us for our call on right? The T2D Knowledge Portal platform webinar workshop. We do these now bi-monthly, and the goal is to actually sort of run through the features and the tools, the data, and sort of the goals and the plans long-term even for the Knowledge Portal platform. We thought this would be a useful environment for people to learn and ask questions. And so our goal for today, my slides would advance. Hold on one second. Excellent, thank you. We have sort of, I'm going to do a quick intro, but not spend too much time because I think I really want to dive into the features and tools. And Maria's going to take you through sort of the T2DKP component about resources for epigenomic annotations and how we interlace those with human genetics data. And then Parul is going to take us through our sister federated node for epigenomics data at UCSD, where they have the Diabetes Epigenome Atlas. And so we're going to have a demonstration of that tool and then how it all connects to the Knowledge Portal. And then we'll have some time for Q&A. The goal today is to try and get to those two topics. So what will you learn today? You will learn how to query integrated human genetic data, layered with genomic annotation for your favorite gene or region, so that we want to go through. We want to talk about what the Diabetes Epigenome Atlas is, so how it came to be as well a little bit, how it's connected with the T2DKP. You'll get that in two flavors, one from Maria and one from Parul at the end. What data does it contain? What types of things can you query? And sort of you can able to ask questions like, does it enhance my understanding of genes areas? Like, how does it help me? And then finally, how might you use it to prioritize functional study, your favorite gene or region? Kind of where we're headed with the resource over time. And if anyone has any questions as we pop in, you can put in the chat line, or we'll save them to the end as well. Future webinars, which we hope to have, bi-monthly, as I mentioned, we're going to have several topics. One is our interactive LocusZoom. We're going to have our Michigan team come and talk about that tool that's embedded in the portal, because there's a lot of features to that you may not know about right away when you log into the portal. We're also going to talk about what types of complications data we make available for query and how we are expanding to metabolic disorders over the next year. I think that'll be an exciting topic in a future webinar. You'll also learn about new workflows as we expand for gene variant prioritization. You also learn about what methods are available and have been run on large-scale GWAS data sets and how you might access those. And then hopefully over time, how you might interact with the T2DKP software stack via the command line, via APIs, so you can run your own tools and methods. And then finally, how do we represent data residing in geographic locations? This is because of our federated node at EBI. We'll talk about that in future webinars. So that's some things to look forward to. What events are coming up that you might care about? We have a large production release mid-month, next month, for ASHG, more data, enhanced features, and a new look to the portal. So that's going to be exciting. New videos, if you bless you. New videos, if you love our videos on our YouTube channel for DGA, LocusZoom, data intake features coming. And then at ASHG, which is kind of our premier event of the year, we have a booth, a platform chart featured Marcin, and a workshop where Jesse Engert and Jason and a few of us on the team are going to talk about association to function resources as the portal develops. So that's exciting. Hopefully you can tune into those. And if you want information, just email me or Maria or the help at help at T2D Genomics. So just before we dive into sort of Maria and Parul's presentation, I want to remind you what the simple idea that got us here. It is the idea of the portal's basic idea at its core is making genetic and related genetic data broadly accessible and useful, hopefully to have an impact on disease, specifically diabetes and its complications. That's why we're here. That's what we are set up to do. And that's what the platform has been created to do over time. This is what it looks like today. We're not going to talk about, we've had other webinars to kind of think through the high-level features of the portal, so we're going to focus on one today. And really, what the portal does is take advantage of an opportunity and challenge. And over the past couple of years, we've done a great job, I think, I can boast if I like, to bring together human genetic data from the diabetes genetics community and make it publicly available. And as a result of that, and all the sequencing studies, genotyping studies going on, about 240 loci have been identified for type diabetes. We've made those available to you, but we must go further if we want to actually make this thing useful. We need to implement and integrate tools and methods and data from the multiple research areas that are needed in order to go from variant to mechanism to therapeutic target. So in order to do that, we really need integration with other federated sites that have domain expertise in other data, particularly epigenomic data. So today we're going to talk about how the Knowledge Portal and the DGA interact and represent data to you that will be useful in that endeavor. And so what powers our whole platform, just to give you a sense of like under the hood, what we look like. When you see the portal, there's, I would say, 6 components. One is our data ingest, which happens at the DCC, where we can bring in summary level statistics, individual level data from genetic data, process it in the best practices of the field, represent those in the portal. We can also bring in summary level statistics, aggregate those, and have a lot of them, and that's primary, our heavy foundation of data, and those can be represented in the portal. But most importantly, we need to be able to run those through a pipeline that allows you to have them run on with computational approaches and methods that are useful for gene and variant characterization. That's this Neo4j knowledge graph where all the results from these computational approaches are stored. But we can't be the experts in everybody. We get very lucky that we have 2 amazing federated sites, one at EBI, which is not the subject of today, which is for genetic data that cannot leave Europe. They're federated with us, and everything they do is sort of like a mirrored version of us. And then if you're on the portal, you see the data in an integrated fashion, which I think is fantastic. Then the other component is DGA. So this is Kyle Dalton and his group are funded to build a federated node for epigenomic data. And that's what you're going to hear about today. You're going to hear about that resource as a standalone resource and how it's connected to the portal and how we represent data. We thought we'd start with actually how it currently is represented in the knowledge portal and then wind you back to how you might use it as its own standalone resource with Google taking over. That's sort of like the gut. So with that, I'm going to turn it over to Maria to walk you through how you can visualize these data in the portal today.
Okay, and so I usually start with this slide just to remind new people that if you want to access any of the knowledge portals, you need a Google account. And what I'm going to focus on the T2D Knowledge Portal today, obviously, but there are epigenomic data in the other portals as well, so you can access this data in a similar way to how I'm going to describe it for the T2D portal. Okay, and so today you can find epigenomic annotation basically in 3 different places in the Type 2 Diabetes portal. On the variant page, on the gene page, and in the tissue focus table, which I'll talk about in a minute. So I'm just going to go pretty quickly through these and then hand it over to Carl. So the types of epigenomic data that we currently have in the Type 2 Diabetes portal are 2 right now. And they're both derived from this paper, Varshney et al., 2017. And so the types of data are, first of all, chromatin states. And these are predictions of the regulatory potential of a given region of DNA. They looked at histone modifications in many different tissues and applied, I think they used machine learning, I don't know, they came up with an algorithm that categorized the genomic regions by their regulatory potential. For example, active enhancers or transcription start sites, and there are about 13 of these states, I think, that we have in the portal right now. And then the other type of epigenomic data that we have is ATAC-seq tracks. So these show open chromatin, and open chromatin, of course, indicates the regulatory capacity of the genomic region. It means that the chromatin is accessible to RNA polymerase and transcription factors and all those important proteins. And we have these tracks right now for 4 different tissues: pancreatic islet, liver, adipose tissue, and skeletal muscle. So the variant page, one place you can access these annotations. And on the variant page, there are 2 places where you can see them. There's a section called Epigenomic Annotations, and then they're also displayed in LocusZoom. So in the Epigenomic Annotation section, there are basically 3 different displays that I'll walk you through. So first of all is an interactive table. And this just lists everything. It lists all of the chromatin states. So we're starting on a variant page, a page for one particular variant, one genomic coordinate. So this table lists all of the chromatin states that overlap that coordinate, and the tissues in which they are found, and then the start and end coordinates of the chromatin state. And you can filter this table. You can filter by chromatin state, and just choose the chromatin state that you want to see, or you can filter by tissue, and then the table will narrow down to just that state or tissue. The columns are also sortable. So also in this section are a couple of graphical ways to look at these data. So I apologize, it's a little bit small, but so this one graphic shows the chromatin states that overlap this variant by tissue, and they're color-coded, and they're also, the names are listed at the bottom here. So for example, this little orange box at the bottom shows that this variant is in the region of an active enhancer 2 in, for genomic view of the chromatin states. So it shows their extent in a kind of a simplified genomic map relative to the position of the variant, which is that line going down the graphic. So again, this is by tissue, and the color key tells you what the chromatin states are. So that section just really gives you everything there is to know about the chromatin states in the area of this variant. Now, LocusZoom plot on the variant page, if you click on any variant, any point in the LocusZoom plot, you'll open a tooltip, and then if you click on Expanded View, you get a view of the epigenomic states around that variant. So before, we were looking at the epigenomic states around the variant of the page, but here you can expand your view to see any variant in the region and look at what epigenomic states, chromatin states are overlapping that variant. Okay, so that's it for the variant page. Then on the gene page, there are epigenomic tracks, which we've talked about a little bit in these webinars before. So just to orient you, on the left is the whole gene page. I know it's tiny, but the top variants tab, which is usually the one that opens by default, is where the LocusZoom plot and the epigenomic tracks are displayed. So here's a larger version of the epigenomic tracks. And so when you hover over these tracks, you'll see some buttons, and one of them is show at-seq reads, and one of them is split tracks. So I just want to show you how those work. So split tracks opens up that ribbon of color to separate the chromatin states. So now you can see where each state is, and there's also a key to tell you what the state is. So like the dark green is weak transcription, for example. And the show at-seq reads opens up a graph like this that shows you the reads in whichever tissue you selected. And of course, I'm not showing it here, but both of these displays are lined up with the LocusZoom plot, so you can see exactly where these peaks and these states are relative to the variants of interest in the plot, or the gene of interest. Okay. Also on this plot, on the gene page, you can add any tissues you want. So this tissue pull-down menu at the top of the plot here lets you select any tissues. You can select you can add as many as you want individually, and so you can get a whole line of the tissues of interest underneath the LocusZoom plot. Also, for any variant in this plot, you can find the tissues that have overlapping enhancer regions. So over here, so we've chosen a variant, clicked on it to open the tooltip, and then if you click the tissues with overlapping enhancer regions button, it will find all the tissues in which enhancers overlap that one position. So for example, here's on the left is the default LocusZoom plot. And then when we click the tissues with overlapping enhancer regions, we get a whole bunch. So I didn't have room for all the tissues. There are many tissues in which enhancer regions overlap the position of that one variant. So this is a quick way of seeing which enhancers are and which tissues could be relevant for the variant that you're looking at. Okay, another place on the gene page where you can see epigenomic data is on the credible sets tab. And I've gone over this before. I'm not going to do it in too much detail, but on this tab, there is a table showing all of the variants in the credible set and color display at the bottom. Lost my mouse here. Here it is. Okay, this color display at the bottom shows which chromatin states out of these four shown at the bottom overlap the positions of each variant in the credible set. And so this might indicate to you that some variants in the credible set are more interesting than others. For example, here these have overlap adipose tissue enhancers, so that might be more relevant for diabetes than the ones that overlap, enhancers in the brain. So that's the default display, but you can also add more tissues to this display. So there's a pull-down menu that lets you select chromatin states, and So these are the 4 that are selected by default. But if you select all of them, or any combination that you like, you'll get a lot more tissue, a lot more data that shows the different states in the different tissues. Okay, and finally, the tissue focus table is the last place right now where there are epigenomic data in the T2D portal. So this is a table that displays the results of several different methods. But the one I want to focus on is GREGOR right now because it uses the epigenomic data as an input. So GREGOR takes the chromatin state and other annotations from DGA, and it combines them with bottom line ancestry-specific genetic associations that we generate here by meta-analyzing all of the GWAS summary statistics that we have in our database. And the algorithm accounts for sample overlap. And so GREGOR comes up with a p-value that describes the significance of association between a tissue and trait. And the p-values are specific to chromatin state and ancestry. So you can interact with this table in the following way. In the GREGOR column, there's this little gear icon, and if you open it up, you can filter the annotation. So it will show you which chromatin states, when calculating table, you adjust those, and I'm not showing the whole list here. There's a long list of chromatin states. It's more than the 13 we have in other places in the portal. It's many, and so you can adjust this any way you want. And when you do, the p-values update dynamically to show the results using those chromatin states. All right, and just a little look into the future. We hope very soon to get more attack-seq data into the Type 2 diabetes portal. We're also going to have transcription factor binding sites, high-C chromatin confirmation data, and also the attack-seq and the high-C data are going to be integrated into that GREGOR calculation that we just talked about. And also in the GREGOR calculation, the tissue types and cell lines will be harmonized. Right now we have a long list of tissue types and cell lines from different projects, but some of which aren't really the same thing, but they're showing as opposite independent now. But Kyle is working very hard on harmonizing them, so it'll be a simpler and more relevant list. All right, I think that is all that I had to say in my part. Let's hand it over to Pearl.
Hi guys, can you hear me clear?
Yeah, we can hear you.
Okay.
And can you just say next slide when you want it? I'll do that.
Sounds good. Thank you, Maria. Professor Kyle Chromatin is the PI on Diabetes Epigenome Atlas. Hi, I'm Parul Kurtarkar, and I'm the software developer at DGA. We are also joined by Ying San, who is our data manager. Next slide. So most of the variants that affect diabetes risk are non-coding variants that regulate gene activity. And identifying the genes affected by non-coding risk variants is crucial for understanding the molecular pathways dysregulated in disease. And these can thereby inform discovery of therapeutic targets. Next slide. Epigenomics is one of the tools to interrogate the function of these non-coding variants. And gene regulation through detailed study of chromatin structure and cis regulatory elements. And that's where study of epigenome and non-coding variants comes into picture. Next slide. So here we demonstrate the activity of regulatory elements is often restricted to specific cell type, and hence it is important to interrogate and map regulatory elements in individual cell types within diabetes relevant tissues, as Maria has also demonstrated in her presentation. Next slide. So this is a figure from one of our recent publication. In this manuscript, we provide a roadmap of how single-cell accessible chromatin data derived from disease-relevant primary tissues can be utilized to define cell types, cell states, cis-regulatory elements, genes involved in genetic basis of complex disease such as diabetes. I'm not going to go into detail of this figure just in favor of time, but if you have any questions later, we can talk about it in detail. Next slide. So based on this, on the premises of these previous slides, emphasizes the need for epigenomic resource to host data from diabetes relevant tissues and cell types. So Diabetes Epigenome Atlas is a data science platform for understanding non-coding disease variants. This is a cloud-based resource that is based on open source software developed as part of ENCODE to host and disseminate human epigenome data relevant to studies of diabetes. As part of this resource, we have collected and reprocessed sequence data from thousands of epigenome assays and developed tools to intersect genetic variants with relevant epigenome annotation. One of the highlights is we have also developed platforms within our system to enable integration with other resources such as T2D Knowledge Portal. Next slide. So this is the landing page of DGA. This is the landing page of DGA, and via the data menu that's on the top left corner, you can access the matrix view of epigenomic assays and annotations. The site can also be queried for key terms such as assay names, annotations, tissues, and publications that we have in our site. In addition, you can also query genetic variants for overlap with relevant annotations and annotation data files. Next slide. So this is example of how we have encapsulated and integrated relevant epigenomic assays and annotations relevant to diabetes into DGA. We have assays and annotations that capture cell type and tissue-specific regulatory elements, affected genes, gene expression levels, and variant information. Next slide. In addition to the experiments and annotations, I'll also be on later slides, we'll also be showing our experiment page and annotation page. But on these pages, we also have metadata such as donor information, biosample, external references from where we collect the data, labs that generated these data, any publications that are associated with the annotations or assays, raw files and process files that can be downloaded individually as well as in batch, along with processing steps that are used to generate these process files. So this is basically a record keeping part of the entire system. As Maria finally mentioned, in order to integrate with federated sites, in our case, T2D Knowledge Portal, the metadata has to be standardized. And we use standardized ontologies such as Oberon, cell ontology, assay standardized EBO or assay experimental ontology for easy query and integration with external resources. Next slide. So you can access the annotations and assays that we have in DGA through the matrix view that can be accessed via the data menu on the top left corner. Next slide. So this is the matrix view of the experiment. It shows different assays that we have on our site, and you can filter them based on the assay of interest, the biosample, the data files that are available with regards to the assay. Next slide. The annotation view is similar to the assay view, where again you can annotate on based on the annotation type, the files, the biosamples, the cell type. Next slide. So this is our experiment page, and on the landing page there is a database search bar where you can put a key term. So in this case, if you query FOXA2, it will give you all the assays that are associated with FOXA2. Next slide. So this is experiment metadata page. You have all the metadata that was previously mentioned associated with an assay, along with the attributions of where the data set was generated, and then the biosample information, replicates associated with the assay, raw and processed files that you can download individually from the experiment page, and then also links to the cell browser, where if there is a single cell assay associated with the data set, you can view it on a cell browser, and a genome browser, which is at the moment in beta state, where you can visualize the signal tracks associated with that assay. Next slide. Next slide. Oh, yeah, sorry. Yeah, so annotation page is similar as assay page. So this is the variant search. So you can query for the variant of interest, such as the RSID or single base pair region, through the landing page, or there is a tool menu on the left-hand menu of the site. And this will generate annotation. This will return the annotations and annotation files that overlap your queried variant. This also gives you peaks that intersect and information such as coordinates where it intersects, the state for the given annotation type, and the value or the score associated with the annotation. Next slide. So you can bulk download both the raw process files, metadata file, individually through the annotation and assay page, as well as through the bulk search. You can also download the metadata and these files in bulk. Also, you can interact with our interface through API that provides a programmatic access to metadata and data files. Next slide. This is, I'm kind of quickly going to iterate what Maria mentioned, the new data sets and features, upcoming features in DGA, and that would be also integrated into T2D Knowledge Portal. So we are going to have additional chromatin state annotations, accessible chromatin annotations from complication tissues, and cell assays. So right now we have diabetes specific tissues, but we are also working on adding diabetes relevant complication tissues. And then because as mentioned in the very first slide, it is important to have cell-specific assays will be including these on our site as well. In addition, we are going to have target gene predictions, variant effects, and all of this data would also be integrated into T2D Knowledge Portal. Next slide. So this is our genome browser. It is one of the upcoming software feature. If you have account with DGA, you can access it. It's in beta mode. It's an embedded browser, genome browser, to visualize signal tracks from individual or multiple assays and annotations together. Next slide. For single cell assays, we also have UCSC single cell browser, which is again in beta mode for single cell data. In this cell browser, each point is a cell, and it is clustered by cell types. You can click on or hover over the cell type for metadata information and search for marker peaks. Next slide. So this is another tool that is in development, and this tool would be integrated into T2D Knowledge Portal. It is a graph-based interactive visualization of variant target gene predictions that would be because how a non-coding variant or a variant relates to the target genes is crucial to understanding the etiology of the disease. This tool is going to be quite informative visualization, and as mentioned, it would be available both on DGA and into T2D Knowledge Portal. Next slide. So then this slide relates to one of the very first slides that we saw, the data that is available and encapsulates data in DGA, and how it will be integrated into T2D Knowledge Portal, which is the genetic platform for T2D. So here we as Maria mentioned, I'm quickly going to dash through things. The epigenomic annotation table on the variant search page on the knowledge portal, the credible set enrichment, the credible set annotations on gene locus page are some of the places where the data would be integrated and hosted. Next slide. Okay, so again, these are Professor Kyle Lawton's slides. He's the PI on the project, and Ying Sun is our data manager. So if you have data sets that will leverage our system and the T2D Knowledge Portal, please get in touch with us. If you have any comments, feedback, do get in touch with us. We are very receptive. And our lab believes that discoveries happen through collaboration. Hence, we would like to acknowledge our amazing collaborators at the T2D Knowledge Portal, the AMP Consortium, our collaborators at UCSD, and the ENCODE project. Thank you.
Thanks, Parul. And we have a few acknowledgement slides ourselves. This is our team. And the whole AMP-T2D partnership is a very large group, people who work together. And these are the more specific teams who work on various aspects in the federated nodes. Thanks to everyone. Now, questions? They unmute themselves. We give them power. Okay, good. Any other questions we have from everyone?
can see anything in chat, so feel free to speak up if you need to. Quiet group today. Well, actually, you know what? If we end early, that's fine. So we're going to have our next one for this is when, Allie? Do you remember? November. Great. So, and it's noon on Thursday, so I don't remember which Thursday it is. But we'll, I think it's 14th. It's the 14th of November. So perfect timing. And then also we mentioned there's several events at ASHG. So you can come talk to us at the booth if you're at ASHG. Rule and Kyle and others will be there, whether they like it or not, to come talk to you about it. So hopefully they can speak to them there. Also, you know, in the coming year, one thing to think about in the future, we hope to integrate DGA more seamlessly in this T2DKP, as Parul was mentioning, with visualizations that will be exposed. We have visualizations that we're working on in partnership as well to prioritize variants and genes, as Parul mentioned. So you'll see those in the coming months as we go through. As Parul mentioned, we're also interested in any other data sets you think would be relevant for type 2 diabetes and its complications. And also thinking beyond just diabetes, metabolic disorders as well, things related to cardiovascular endpoints and whatnot are also valuable to this resource. So if you have any other questions, you can either post them to us afterwards, or we'll pause one more minute, or otherwise we'll give you back 20 minutes of your day.
Can you hear me? This is
a quick question here. For some of the visualizations, especially in the first part of what we saw today, for putting together a figure for publication, what would be at a level where you could just take a figure or part of a figure right from this, or would it be standard to sort of take the code somehow that it generates this and be able to personalize it?
So to answer the question for a publication, we would like to use them actually, because we think that's a great thing to actually sort of, make this resource useful and just sort of cite a portal or DGA. But if it's something you want to use more for computational things, certain ones of these are available for command line access, depending on what you're interested in. So both is actually, so if you were thinking of something on a processing basis, yes, we do make it available. We do work with you actually to help implement if you want to it. But the standalone, figures and visualizations are meant to be used in papers.
And also, just a technical note about generating a figure. For LocusZoom, there's a button on the upper right of the slide that says download SVG file. And so that will give you a nice publication quality file of the LocusZoom file you're looking at.
And we'll probably do those for more features that I think will be more relevant, like particularly from when Parul was showing the different nodes. I think those are pretty nice. Parul, do you have a means to download those, or how do they work now on DGA?
Yeah, I mean, you can, if those are signal tracks, you can download the signal tracks. So the Cicero plots that were shown for the locus and accessible target genes, those are not downloadable yet. There is another tool that we'll be working on. Yeah, so that's where we are at the moment. Yeah, but as we develop visualization tools, will ensure that you can download them as PNG or JPEG files for publication.
Great. Perfect. That answers the question. All right, everybody, thanks. We have a fair number of people on the line, so that's excellent. So we'll see you in a month and a half, roughly, and we'll see you all at ASHG, hopefully. So thank you to our presenters. Thanks, Maria. Thanks, Kyle, who couldn't be here, and thanks, Parul. We'll see you all next time.